Hello everyone, my name is Muhammad Hadi Ali. I'm a 6th year med student at Beirut Arab University, and today we're going to talk about the complement proteins in inflammatory response. If you have any question, you can email me at my email over here. So what is this system? The complement system is made up of a bunch of proteins that are liver synthesized and float around in your blood. Think of them as a team of superheroes waiting for a signal to spring into action. When activated, they perform three main jobs. They directly lyse pathogens via the membrane attack complex, they enhance phagocytosis through opsonization, and they amplify inflammation via chemotaxis. But why does it matter? The complement system is super important because it provides a rapid and effective defense against microbes. It acts as a frontline surveillance mechanism. It can eliminate threats on its own and also works with other immune cells to create a powerful defense. A common mistake is underestimating the multiple functions of complement proteins or confusing the triggers for its different activation pathways. As you can see, the main components of the complement system are complement proteins. These are individual proteins labeled with the letter C and then numbered from 1 to 9, and there is also another protein called factor B. All of these proteins, they participate in the complement cascade. And we have three main pathways: the classical pathway, the alternative pathway, and the lectin pathway. We're going to discuss each pathway one by one. Now this is an outline of what we're going to explain: the pathways of complement activation as we mentioned earlier, and the key functions or outcomes of complement activation, which are the membrane attack complex formation, opsonization, and enhancing inflammation, which are the three main jobs the complement system performs. First, let's talk about the classical pathway. This pathway is antibody mediated. This means it starts when an antibody which has already met a pathogen binds to an antigen on the pathogen's surface. This binding activates the first complement protein, C1. A cascade then follows. C1 activates C4, which activates C2, and then they end up with a complex called C4b2a, which acts as a C3 convertase. The C3 convertase splits the complement protein C3 into two parts, C3a and C3b. C3a diffuses away, and C3b binds to the pathogen's surface. Then the cascade continues, leading to the formation of a C5 convertase, which also splits C5 into C5a and C5b, and also C5a diffuses away while C5b binds to the pathogen. Now C3a and C5a, they are the small complement fragments. They act as powerful chemotactic agents and anaphylatoxins, enhancing inflammation and attracting white blood cells. Next is the alternative pathway. This pathway is super fast because it's antibody independent and involves direct recognition of pathogen surfaces. It's initiated when the complement protein C3b directly binds to an antigen on a pathogen's surface. This direct binding also leads to formation of a C3 convertase and then a C5 convertase, continuing the cascade to C5b. The final pathway is the lectin pathway. This one is also antibody independent. It starts when the mannose binding lectin, which is a protein, binds to mannose sugar molecules found on the surface of certain bacteria and other microorganisms. This binding activates associated proteases, which then activate C4 and C2, forming a C3 convertase and continuing the cascade to C5b. Now here's a visual of the three pathways of the complement system. The classical pathway is antigen-antibody dependent. On the other hand, the alternative pathway and the mannose binding lectin pathway aren't. All of them, they start differently, but at the end, they end forming C3 convertase, resulting in C5 convertase, resulting in the lytic pathway, which is common between all the three different pathways, which is C5b, C6, C7, C8, C9. And finally, they make this complex, which is called the membrane attack complex, which directly lyses pathogens. Now for the key functions and outcomes of the complement activation, first, as we mentioned earlier, we have the membrane attack complex formation. The complement protein C5b, C6, C7, C8, and multiple C9 units assemble to form the membrane attack complex. It is a pore-forming structure assembled by complement proteins that we just mentioned, which inserts into pathogen membranes causing lysis. The MAC inserts itself into the cell membrane of the target pathogen, for example a bacteria, creating a large channel-like pore. This pore disrupts the pathogen's membrane integrity, causing fluids and ions to rush into the bacterial cell. This influx of fluid leads to the pathogen swelling and ultimately it undergoes lysis. Second, we have opsonization, which is in other words enhanced phagocytosis. The C3b protein we talked about earlier acts as an opsonin. An opsonin is a molecule that enhances phagocytosis throughout coating antigens. When C3b binds and coats the surface of a pathogen, it makes the pathogen more recognizable and, let's say, tastier for phagocytic cells like macrophages and neutrophils to engulf. Phagocytes have specific C3b receptors that bind to the C3b coated pathogen, significantly enhancing the efficiency of phagocytosis and leading to the pathogen's destruction. Finally, we have enhancing inflammation through chemotaxis and anaphylaxis. We talked earlier about C3a and C5a. We said they are small complement fragments. They are powerful chemotactic agents. They attract and guide more white blood cells like neutrophils and macrophages to the site of infection and inflammation. C3a and C5a also act as anaphylatoxins, directly activating mast cells and other cells to release inflammatory mediators, thereby further amplifying and enhancing the inflammatory response. Now let's recap. The complement system is a vital component of your innate immune system. It provides a rapid, powerful, and non-specific defense against invading microorganisms. Its ability to directly lyse pathogens through the MAC complex, enhance phagocytosis through C3b coating the pathogens, and promote inflammation through C3a and C5a makes it a critical first line of defense. It has multiple pathways which are triggered by antibodies, pathogen surfaces, and mannose, but all pathways converge to activate C3 and C5, leading to the same crucial effector functions. It surely cooperates with other defenses when it enhances phagocytosis and contributing to the overall inflammatory response. And that's the powerful complement system. It's an incredible example of how all the parts of your immune system work together to keep you safe and healthy. Thank you for watching.